This morning, we'll be watching a video first to recap what the story of Gregor Mendel from last week. And then we'll introduce a few more genetic vocabulary before we breed pigeons in a computer game. Every year, right, a pair of pigeons will lay 2 eggs at the aircon ledge. Every year, you know. Yeah, then I'll just watch them go up. Yeah, and believe me, I thought that all babies were cute. Pigeon babies, right? So ugly. Like, so tempted to push them off the edge. But like you thought that babies, pigeons would look like little chicks, nice and furry. Not. They just perpetually look wet. Like, they perpetually look slimy all over, yellow in color. It does not get any better. Okay, so a summary from last week.
These days, scientists know how to inherit characteristics from their parents. They're able to calculate probabilities of having specific traits or getting a genetic disease according to the information they have from their parents and family history. But how is this possible? To understand how traits pass from one living being
Wait, I
to
want to
descendants,
check if going speed slower would sound weird.
we need to go back in time to the 19th century.
Oh, okay. Start from there. Okay, this person speaks at lightning speed. Okay, that's not very slow. Okay.
These days, scientists know how to inherit characteristics from their parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous gene, where both alleles are identical, and what we call a heterozygous gene, where the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase Y always overpowers lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the 3 possible genotypes, showing the 2 possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations. Round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett spread too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
Okay, so that's a quick recap of a story that we shared last week. Some of you are uniform, huh? Oral, is it? Right. How many chromosomes do we have in each cell? 23? No, we have 46. Okay, we have 46 chromosomes in every single cell of yours. Okay, we have 46. But actually we have 23 types of chromosomes. Right? In other words, you look at all these lines that drawn here, they represent chromosomes. Actually we have a pair of each one. So let me draw in the pair. The homologous pair. We have 23 of these pairs. Notice that homologous chromosomes, they tend to be same in length. That's because they contain the same genes. So 23 of these pairs. The last pair actually determines your gender. So the last pair of chromosomes are your sex chromosomes. For example, X or Y chromosomes. Depending on the pair, you get different genders. So this is the first thing to establish. 46 chromosomes, 23 pairs of homologous chromosomes. We introduced the words genes and alleles to you. Yeah? Every chromosome in your body contains genes that encapsulate who you are as a person. For example, maybe on chromosome 1, at this particular point of the chromosome, you have a gene that determines your eye color. Perhaps at chromosome number 4, at this particular point over here, you have a gene that determines your hair color. Maybe at this point over here, at chromosome, you count, you have a gene that determines your the size of your nose. Okay? But we have pairs of chromosomes, right? So each pair of chromosomes has a corresponding allele. You could have the same allele or the different allele. So for example, perhaps at this point over here, you have the same allele. Maybe at this point over here, you have an allele that determines a slightly longer nose. Maybe at this point over here, you have an allele that determines red hair color. It is the same gene at this point between the pair, but the form of the allele may be slightly different. It is the combination of the alleles that determines what you see on the surface. And we learned last week that depending on whether the allele is dominant or recessive, could determine what you see on the surface. Let's say red hair is the dominant allele. You have this pair, this combination, on the surface you will have red hair. Let's say short nose is the dominant allele. In this form, if you have both, then your nose will be short. Let's say you have the same alleles for both, for eye color, black. Then on the surface, your eye color will be black. In our study at the JH4 level, every time we give you puzzles to solve, we usually ask you, we only solve puzzles involving just one pair of chromosomes. We never ever ask you to solve a puzzle involving many different characteristics across many different chromosomes. That's too complicated to solve. Right? So always just one pair. That's the good news. So, some terms that we learned last week. We learned about dominant and recessive. The idea of dominant is that if in a pair of alleles, we have the dominant and recessive, the dominant will show on the surface. Right? for recessive allele, it can only show itself if you have a pairing where both are recessive. Yeah? Of course, if you have a pairing where both are dominant, then the dominant trait will show on the surface. So that's what we learned last week, dominant and recessive. Today, we're going to introduce a few more terms. Homozygous, heterozygous. The word homo means same, hetero means different. So I'd like to introduce a new way to describe these pairings. You could say that this is homozygous pairing. This is also a homozygous pairing. This is a heterozygous pairing. Yeah? So homozygous, heterozygous is used to describe what's the combination of the pair. Two terms, genotype and phenotype. The word geno refers to the genes, or rather the combination of the alleles present. Phenotype, pheno represents like your characteristics on the surface. So, how might I use this in a sentence? I may ask you, what is the genotype of a pea plant that is yellow color? Could say, oh, this particular organism has a homozygous genotype. So, genotype describes the combination of the pair. Okay? So, there are several ways you can answer the question. If someone were to ask you what's the genotype of an organism, you could say that the organism is homozygous dominant, you could say that the organism is homozygous recessive. What that means is that this organism has same alleles for the dominant allele, or it has two of the same recessive alleles. Yeah? So I say again, if you didn't catch me, if someone were to ask, what is the genotype of this particular organism? My reply could be, this organism is homozygous dominant. This organism is homozygous recessive. Instead of saying this organism has one dominant allele, has two dominant alleles, or this organism has two recessive alleles, quite a mouthful, right? If you can just say homozygous or heterozygous, it's much more succinct. Phenotype describes what appears on the surface, depending on the genotype. For example, one might ask you, what would the phenotype be for an organism that is homozygous dominant? I'll say again, what would the phenotype be if the organism is homozygous dominant? Your reply would be that the organism would be yellow. If someone were to ask you, what is the phenotype of an organism that has a genotype that is homozygous recessive? Well, it would be green. Or if someone were to ask you, what is the phenotype of a plant that has a genotype that is heterozygous, your reply would be, it is yellow. So if someone were to ask you what's the phenotype of an organism, they're just asking you what's the characteristic on the surface. If someone were to ask you what's the genotype of an organism, they're asking you, hey, what's the combination? Okay? So these six vocabulary you use very often when you want to solve puzzles. If you want to explain yourself and justify why you think this lady will have this genotype or not, these six will often be used. So I'm just going to write down in each instance what color it will be. It will be yellow. Okay, assuming we're looking at the P example, green, yellow. Okay, but phenotype describes the characteristics that you see. Do you know your blood type? Do you know your parents' blood type? Because actually, if you know your parents' blood type, you know your blood type, you can now write down your genotype. Do you know your genotype or blood type? For example, my parent, one parent is blood type B. One of my parents, the blood type is, the phenotype of my mom is blood type B. The phenotype of my dad is blood type A. The phenotype of me is blood type O. What is my genotype? My genotype would definitely have to be homozygous, recessive, yeah? Or another way you could say, I'm homozygous for the O allele. Okay. So this is how I can use all these terms in a sentence. So actually, if you want to know your genotype, all you really need to know is your own and your parents' phenotype. Then you can calculate. Okay, so these 6 terms. With these 6 terms in mind, we'll now play pigeon game. Okay, in the pigeon game, I would like you to figure out one thing for me. What's this? Oh, 6 chromosomes, I wrote there. Okay. So, in this pigeon game, I just have one question for you. Can you find out okay, later on you'll see these 2 alleles. You'll see crest allele and no crest. You understand what I mean later on when you see the pigeons. I want you to find out which allele is dominant. And you must tell me the evidence as you play the game. As you play the game, what's evidence that gives you away that one of these are the dominant allele and one is the recessive? Okay? If you can find out, then you broadly understand the concepts of inheritance. Okay, this is what the game looks like. If you were to access the game, you can go to number 2. You can play together. It's quite fun to solve the puzzles together. This is what you'll see. Oh shucks, it will take ages. You can play on your phone, you can play on an iPad. I would like you to play up to level 5. Okay, play level 1 to 5. I want you at every level to earn 3 golden eggs. Okay? If you earn 1 or 2 golden eggs at each level, means you have not done it to the best of your ability yet. How do you earn 3 golden eggs? By mating the pigeons as few times as possible. Okay? The game goes something like that. They will ask you to create a baby of a particular phenotype. Your goal in each level is to find the right pairing of parents to mate each other to create this baby. The less parents you use, the more golden eggs you will earn.
You've probably seen this guy before, maybe up on a wire or flying around town with about 100 of his friends.
I will play the front part. You hear the narration. Once yours is loaded, you can start playing.
It's a pigeon, or more properly, a domestic rock pigeon. Domestic rock pigeons come in different colors, patterns, shapes, and sizes. In all, there are over 350 breeds. People like to breed pigeons with specific combinations of characteristics. Pigeon fanciers show their birds at fairs. They fly their birds in races where the winners earn million-dollar prizes. Some even write books. The first thing you need to know about pigeon breeding is that, just like people, pigeons can be male or female. Sex is determined by sex chromosomes. In people, they're X and Y. In pigeons, they're Z and W.
Notice, for male pigeons, their genotype ZZ. For male humans, the genotype of male pigeons is ZZ. The genotype of male humans is XY. Yeah?
Your first puzzle is to breed a female pigeon.
So the task goes something like that. At every level, you'll be given pigeons. You will choose pigeons you want to get them to mate. You'll get to choose the chromosome or the allele that will go into the next baby. And then you'll hatch the baby. Okay? There are 5 different levels. This is the easiest level. As the level progresses, there will be more pigeons to choose from on the wire that you can play with. I'd like you to figure out which allele is the dominant allele and what's your evidence for it as you play levels 1 to 5. Okay? Now you have some free time to play the game. You can play together, especially if yours is taking a while to load and your friend has got it ready. I don't know why some iPads will reload. Like they play a little bit and then reload. And some people don't have that problem. Oh, you're trying to solve it. Those that understand the game and you manage to solve up to level 5, you go on if you want to, as your friends are trying up to level 5. It gets harder and harder to solve the puzzles. I just need you to answer that one question for me on the whiteboard. Actually, there's an app version of this game also. So if you have access to App Store and you feel like playing all the way to end without internet access, right? There's an app version of this game. Quick tallies for the crest, you know which one is dominant. How do you tell it's dominant? Think which one is dominant, sir? Like how you tell? Okay, so actually it's good that you manage to identify which word to look at. It's always the heterozygous but always the reverse. And then the same logic applies for a lot of the other values of them. But just that now we have to do it very fast. If the level gets harder and harder, I don't know how far you can go. I think I like 20 plus levels and then no, it's not by luck. It is there's logic to it. So for example, what crest when you figure out yet which value is dominant. Because right, actually if you know which value is dominant, then you can strategize which values you want to use. Yeah. So the first few levels of but actually when we figure out play the game, I hope that it's not by chance. There is logic to this puzzle. Wow, we progress so far already. Okay, now I have a question. So, press and don't press the lead. Which one? Since you progress. cut playing our games at 20. We'll pause a while to consolidate our learning. This particular gene is very special because neither the no slipper or the slipper are dominant over each other. Notice that when both slippers are present, you get nothing. Then when it's no slipper, you get the slipper. But then when you have no slipper and slipper, it's like in between. So this particular gene behaves a bit different. Yeah, so actually this one is quite tricky. So if you want something with slipper, right? So you have to figure out which one is best. Oh, progress very far. Okay, could you pause the game? When you pause the game, actually you will see what level you are at. Quick pause, then you can see which level you are at. There are a total of 26 levels. Okay, I have to caution you. Actually, beyond level 5, you're looking at senior high content. Yeah? So if you play beyond level 5, you are actually using logic and intuition to understand senior high level genetics. We just need to understand up to level 5. Beyond level 15 is university level. So as I was playing up to level 15, 16 onwards, right? Well, I also don't understand how to play already. But if you use your logic, you can sort of see each gene is inherited a little bit differently. Now I would like to just run through the central question that I asked you. And I think many of you were able to solve this simple mystery. I ask you whether you can tell me whether the crest or no crest is the dominant allele. Can you tell? Then my next question is, what's your evidence? In fact, just from these two pigeons alone, you can already tell which is the dominant allele. Alright? Actually, one pigeon here is more important than the other to be able to tell which is the dominant allele. The one which is, okay, let's use the words on the whiteboard. The pigeon that has the heterozygous genotype. So, if someone were to ask you which is the dominant allele, you zoom in straight for the heterozygous genotype. Because whatever phenotype is showing, then you have an idea of which is the dominant allele. Yeah? So, actually, a lot of you all managed to solve this puzzle without me teaching you this. So, I want to show you that this puzzle solving in this chapter there's intuition to it. Okay, so always aim for the heterozygous genotype. Sometimes you may see this word, you may see people say heterozygote. When you see the word heterozygote, they are referring to the individual. So I'll give you an example how I use it in a sentence. That pigeon on the left has a heterozygous genotype. Or I can say that the individual on the left is a heterozygote. One describes the individual, one describes the individual's genotype. Yeah, so I could say the individual on the right side is a homozygote. Or the individual on the right hand side has a homozygous recessive genotype. So, two ways you could use all these terms. Why did I draw three pigeons? I'm trying to recall what I did. So, if you want to represent these alleles in terms of words, usually we use alphabets. Okay, now I want to teach you how to choose alphabets to use. You see no crest and crest, right? Usually, the allele that is dominant, we will take the first alphabet that describes the allele. So for example, no crest is the dominant allele. I would choose the alphabet N to represent the allele. Then the recessive one, I'll put small n. Usually it's like that. So for example, if eye color, if blue is the dominant over, let's say green, then I will put big B as the dominant allele. Sorry? It's okay. It's okay whether if, let's say it's blue and black, right? Also okay. So I would say, it's not strict. It's not that strict, but this is one way if you don't think so much. Honestly, if you want to use big A, small a, also can. Okay, the alphabet is not that important. Only one thing I would like you to pay attention to. When you choose the alphabets, don't choose the alphabets that big cat, small cat look exactly the same. Give an example. You choose big C and small c, it's not that different if you're not careful. So, I show you other examples. Big O, small o. Okay, big S, small s, not very good. Choose alphabets like this or this. There is a difference. Okay, you can actually choose any alphabet you want. Okay, but in my case, I chose N and small n just to pay homage, just to give honor to whatever the characteristic is. So let's look at the pigeons here. As we said, if you have a pigeon that is heterozygous for the allele, then it has crest, right? The crest was like over here. If it's homozygous dominant, you'll find that it also has the crest. If it is homozygous recessive, then you'll have no crest phenotype. What did I do? Oh, okay, sorry, wrong. No crest. Should have no crest. And then you'll have crest. You see, I don't know why, sometimes as humans, right, we will think one trait is better than the other, nicer than the other, then we will think that is a dominant one. But it's not true in nature. Okay? Even for the peas, right, until now, I keep telling myself the green one is the dominant one. Actually, in reality, yellow is the dominant one. Okay? So always aim for the heterozygote. It gives you lots of clues. Has anyone played up to the slipper stage? Okay. So the slipper stage is a little bit odd. Don't know if you observe that in slippers, I'm going to just go ahead first. Okay, then everyone can see. You tell me which parents I should use.
We inherit two copies, different versions of.
Okay, which parent should we use to form a baby that is male and no crest? Assuming number 1 is on the left, then number 5 is on the right hand side. Which parent should we use? Actually, 1 and 2 are the same, right? No difference. Okay. So let's choose one of the males first. Okay, so which female should we choose? Last one? Okay, we need a male, so we need 2 Zs. Then we need no crest. Okay, and it doesn't matter, you see, the beauty of it is if you know which is dominant, in reality, a heterozygote will do. Don't need to get 2 no crests. Right? So let's hatch this baby. Let's go to the next level. Which one should we choose? There are 4 pigeons here. 1 to 4. 1 and 3. Okay, we know that crest is the recessive, right? So therefore we will need 2 copies of crest. So we need to find parents with the crest allele because it's recessive. So only when we have 2 copies, then we'll be able to get a baby that has crest. The next level is a bit interesting. Is this the simple one? Oh, not the simple yet. Oh, okay, which one should we choose? Okay, this one needs 2 generations.
Yeah.
Okay, so you all still remember how to solve this puzzle? Okay, how to solve that? Okay, we need to make a female with crest first, okay? Okay, so which one should I choose? Like that? Okay, then take the crest. What are you trying to do here? You are trying to create a female with crest. So that, is this what you want? Okay? So it's saying you want to create a female that carries the recessive allele. So that, because we see there's one male up there, right? Then we can play, okay, so this female and this male. Now we can create a female that is crested. Is this correct? Okay. I think the next one will be the
Some
simple
pigeons
one.
have featherless feet. Others have feet
Oh,
with
slipper.
quasi feathers. Breeders call this phenotype slipper. Slipper is an inherited characteristic.
Okay. The interesting thing about slipper is that it doesn't quite follow the laws of dominant and recessive. Don't know if you observed that. Okay? Okay, so from this level onwards, what they are trying to show you is that not every allele interacts in the same way. We teach you the basics, where one is more powerful than the other. But in nature, the truth is actually quite far away from this. Let's look at these pigeons here. At the moment, can you tell which is dominant? Cannot really tell, right? Because there are no heterozygotes, yeah? Okay, so how? I'm just going to create 8 sharks. Oh, okay. Oh, scan this picture. Thank you. Do you want it to distribution? Okay. I'm just going to create a baby that is heterozygote. Okay, then we see what the outcome is. Then we can figure out which is dominant, right? Okay, let's just take this male and this female. Okay, wait. And this female. Then we will create a heterozygous baby so that we can see what's the phenotype. Let's hatch. The heterozygous baby is here. Let's look at the phenotype. You see, both no slipper, you get no slipper. When you have 2 slipper, you get a full-blown slipper. But when you have no slipper and slipper, you get like a half slipper. So, this interaction doesn't quite follow the laws of dominant and recessive. I'd like to introduce to you something interesting. This is out of your syllabus. Huh? Okay, notice like hybrid, like in between. Okay, so some alleles interact this way. We call this way, I'm writing in red because it's out of your syllabus, incomplete dominance. That means to say, if you have both alleles present, neither of them can dominate over each other. And you get an in-between. I give you some examples in nature. For example, if I have a red flower and a white flower, if both alleles are incomplete dominant, then you'll get a pink flower. We've learned one more form of interaction last week on blood types. Do you remember in blood types, if A, B present, that's not incomplete dominance. Okay, this is within your syllabus. This is codominance. I'm not sure if you can see the difference between codominant and incomplete dominant. I'm going to give you an example, a very far-fetched example. Let's say we have cows. Red cows and white cows. Okay, let's say I'm going to put red as the dominant, white as the recessive. So red cow, white cow. If we are following incomplete dominance, if you put red and white together, you will end up getting a pink cow. Okay, you get a pink cow. So, okay, I'm stretching my art skills already. Okay, you will get a pink cow. Okay, but if it is codominant, that means both will show on the surface. What kind of a cow will you get if in theory it is codominant? Okay. So actually, if it's codominant, both will show up. You will end up getting a cow that has red patches, white patches, and red patches. All first cow white. I know this is hypothetical. Okay? Can I? So, blood type more resembles codominant. Because if you have A and B present, your blood type will be AB. Right? It's not in between of A and B. It is both are present. You do not need to know incomplete dominance. It's out of your syllabus. SH, you'll study that. But codominance, you need to know because this is very relatable. It's related to cardiovascular system in it, as well as, this all it's related to. So in it, it's very related to the heart, the cardiovascular system. Because all the blood types, there's actually codominance taking place. So, if now let's put things back into the blood. I think last week we also mentioned it. A is dominant over O. B is dominant over O. But A and B are codominant. This is how I'm just using them in a sentence. You only write it out if you are quite comfortable and fluent with the language. I also love the example of blood types because blood type shows you that for every gene, you don't necessarily just have two alleles. You could have multiple alleles. And blood type is a very classic example. In the example of the pea color, there were only 2 alleles. But in life, actually a lot of genes have multiple alleles. And blood type is a very beautiful, natural example that is very relatable to us. Multiple alleles. Okay, actually I was kidding. Actually the example of red and white cow is real. Okay, so there are cows that are not pink, but you can get a cow that is red and white. If you ever come across this term RON cow, okay, RON cow is a cow that has both red and white. So this is why it looks like. See, this is a roan cow. It has both red and white. This is another example of a roan cow. Both red and white. It's not your NJ red, okay? Sorry, if you're hoping for NJ red. It's more like a brownish maroon. Okay, roan cow. So that means the parents one will have to be totally brown or red, and then the other parent will be totally white. Then you can get a roan baby. Okay? So if you're ever confused about codominance or incomplete, number 1, remember incomplete is out of the syllabus, so you can never need to write it down. Number 2, you just ask yourself, can you have a pink cow? You have never seen a pink cow before. Okay? Okay, so this morning, you downloaded and you played a little bit SLS, you learned a little bit about genetic vocabulary. Okay? Until 12, right? Yeah. In the last part of the lesson, I'm going to teach you how to draw genetic diagrams. Actually, you are very good at solving puzzles at this point. Now I want to teach you one more level. Sometimes the question may ask you to draw diagrams to solve a puzzle. I'd like you to refer to your SLS. You can continue playing the game when you go home. This one, a few times that you continue playing a game, the year case won't catch you. Okay, your SLS, if you go to tab 2, and you scroll down, this is what you will see if you scroll down, blah, blah, blah. Over here. Why I cannot see this? For your SLS, can you see a diagram provided? There should be a diagram with some blanks for you. Maybe let me refresh. You go to this particular section, under Discuss Interactive Tool, then you click on Predicting Offspring. Okay, here it is. Can you find this image? Will you save the image? This is a template that you can always use when you want to solve genetic puzzles. And very, very high chance, I would say almost 99%, we will ask you to draw a genetic diagram. You can also find the template inside your textbook, or just like run through every part of the template. Here is a sample of a puzzle that we may provide you. And the puzzle goes something like that. A pigeon which is heterozygous for the crest gene mates with another pigeon which is also heterozygous for the crest gene. They want you to draw a genetic diagram to find out the probability of getting an offspring with no crest phenotype. I actually think you can solve it without drawing this diagram. Yeah? But the reason why we still get you to draw diagrams at this stage is because it teaches you a little bit about genetics as you draw it out. When you go to university level, nobody draws this diagram anymore. They just use that square to solve every puzzle. Okay? But for our sake, we are still going to learn the basics of genetics. First and foremost, if you are given such a puzzle, I would like you to begin first by writing 2 titles. Parental generation. And slightly below, you'll find another section, you can write F1 generation. And you do not need to write any of this down. The template is there for you. Parental generation refers to the parents. F1 generation, the F1 is not the racing car. F1 refers to, the F refers to filial. You heard of filial piety? So filial means you are the offspring of. So F1 means you are the first generation of offspring. Okay, so that's what F1 means, F1 generation. On the leftmost column, you see many other sub-headers. You will write all these headers down before you begin drawing a genetic diagram. So parental phenotype, then parental genotype. This is the only thing I need you to memorize, commit to memory. The template. Last but not least, gametes. Under F1 generation, you write some more headers. You write F1 genotype. Wow, an exciting lesson. F1 phenotype. Last but not least, expected F1 phenotypic ratio. Now this is the part where you have to fill A. Okay, so you have the text, right, Mr. Lim? First, okay, welcome. This is where you fill in the blanks. The template is the same for any question. You just have to fill in the blanks based on the question itself. Number one, first row you leave blank. Second row, you will write down the phenotype of the parents. Okay, so I ask you, if both parents are heterozygous for the crest, what is their phenotype? No crest, right? Yeah? Okay, so you write no crest, then you put an X. Everything in blue is what you can swap out for every question. No crest, you will mate with, so X represents mating, no crest. What is the genotype of the parent? They already told you a clue. They said that they are both heterozygous. So you will write big N, small n, big N, small n. Which reminds me, at the very top, you need to define. So let N be the allele for fill in the blank, no crest. And N be the allele for crest. Everything in blue, you just solve out for every question you are given, every puzzle you are given. Okay, next step, you will see that you are asked to draw two lines out. These two lines represent, you do not need to write this down, these two lines actually represent meiosis taking place to form gametes. This is compulsory, you must draw two circles under each genotype. The circles represent the gametes, either sperm or egg. Why we draw a circle? Because we want to show you that it's within a cell. You will then write in the allele present after they separate. And now just pause here for a while for you to take out any notes that you want to take out before we start mating the parents to get the offspring. Phenotypic ratio. You can find it from template. Okay, This part I need you to follow me because you see a bunch of lines over there. How to draw it out. All those lines represent the possible ways in which fertilization can occur. There are 4 different ways fertilization can occur. We actually learned last week there's an easier way to do this. Just draw that square, then you can find out the 4 different ways. But the reason why we get you to do this at the beginner level is to help you remember there is fertilization taking place. So this is how you draw the arrows up. Start off with the first gamete, draw one arrow down, and draw another arrow here. Then the next gamete, draw 2 arrows to fill in the remaining spaces. I'm going to use as many colors as possible so that you can see the succession of how I draw the lines. Next, you will take the extreme right, go down. So this is first combi. This is the second combination. I got no more color. Orange. Lift up, open. Damn it. Okay, let's work with blue lah. Okay. So then one more. Okay. This is how the sequence goes, how you draw the lines. If not, you will confuse yourself. You'll end up having 3 lines converging into one individual. You start off from extreme left, followed by the next gamete. And you start from extreme right again, followed by this one. Once done, may I get you now to write down the genotype of each new individual? Write down the phenotype of each new individual based on what you understand of crest and no crest. Can you tell what the genotype would be, what the phenotype would be? If you're unsure if you cross it correctly, you can draw a table to just double check. The very last row is what you can write. They ask you what will the ratio of the phenotypes be. So this is what you can do. You can write no crest is to crest, these 2 dots, the colon. Then write down what the ratio of the phenotypes will be. You have to give a count. Count how many individuals will be, how many each phenotype will be able to do this ratio. So what is the ratio after you calculate? 3:1. So this is how you can write the last row. Then right now, you can answer the question finally. They ask you for the probability. Okay, I know it's very painful, right? You do all this, right? Just to answer what's the probability. See, this box would have done it. So if you get MCQ question, for goodness sake, don't draw this whole diagram out. Just draw the square. But if you were to get a structured question, it will ask you to draw the genetic diagram. Then you cannot draw this square. Okay? Just read the question carefully. Usually we'll tell you up front, draw a genetic diagram. Okay, I'm going to leave this blank, these 2 rows. I expect you to be able to fill it in, whether it's crest or no crest. And what the genotypes will be, whether they are bigams, heterozygous, or if they are homozygous recessive. So, in the remaining time, I'd like you to practice one more puzzle. Okay, then we will check in next week. It's a very simple piece of homework. You could finish it in 5 minutes or 10 minutes. I'd like you to try this puzzle. A heterozygote mates with a homozygote for crest allele. Crest, that means homozygote for crest. Then they say a heterozygous person, pigeon mated with a homozygous for crest. Construct a genetic diagram using the steps below. Then predict the probability of getting a no crest phenotype. I'd like you to practice drawing one genetic diagram from scratch by yourself to solve this puzzle. Yes? Oh, okay, good question. Do you need to draw these dotted lines to box? Actually, it's a fantastic question. You do not need to. I forgot to tell this to the previous generation. So when the first exam scripts came in, right, I saw people drew tables. And then they somehow even very nice thing dotted lines for me. They spent 3 times more time in other classes. Yeah, only put a dotted line so that it can be neater. Okay, but you do not need to put any of the dotted lines. Your genetic diagram should look very clean. Like this. Like below. Okay? Your genetic diagram will look very clean. Less all those dotted lines. Like how all these people are doing it. The thing they do not have are all the headers on the left hand most column. This one not A, no, this one don't have. This one? Bottom. But they put their headers on the right hand side. Okay? So that's just a small piece of homework. You can even do it now. Just to practice drawing a genetic diagram from scratch. Yeah, from scratch. You are expected to be able to draw from scratch. Or no template will be provided to you. The first row. Make sure you define the alleles before you begin drawing a genetic diagram.